Hey friends, and welcome to today's talk about the RT-PCR, which is short for reverse transcription PCR. We apply reverse transcription PCR when we have an RNA fragment and we want to transform it into DNA so that we can amplify it to sequence it. This is, for example, applied when we have to check if a person is infected by a virus. Since many viruses contain RNA, we first have to extract the RNA and with reverse transcription generate DNA, and then we can amplify the DNA with PCR. We use reverse transcription PCR, for example, nowadays in the 2020 COVID-19 coronavirus, when we test patients whether they are infected with the virus or not. In the normal transcription procedure, DNA serves as a template and RNA is transcribed out of DNA. However, in reverse transcription, it is the opposite. We have an RNA template here, and with reverse transcriptase as an enzyme, we can generate DNA out of RNA. And how this is done, I will show you now in detail. So first we split the RT-PCR into two steps. First we have the reverse transcription. And what happens here is that we need the primer to anneal to the RNA template. The second step is the cDNA synthesis, and this is done with an enzyme called reverse transcriptase. Reverse transcriptase binds in the primer region and is now synthesizing the DNA nucleotides, the newly cDNA strand. Now we get to the PCR. This double-stranded cDNA here has first to undergo a denaturation step, so the strands will split and then specific primers can anneal to the DNA fragment here. A DNA polymerase, which is known from replication, can now join the DNA fragment. In most cases in the lab we use so-called Taq polymerase, which is very heat stable. The Taq polymerase can now synthesize double-stranded DNA. If we repeat several different cycles, we can generate multiple fragments of DNA out of one. If one DNA fragment serves as a template, in the second cycle we have already 2 DNA fragments, and in the third cycle we already have 4, and so on and so forth. If you want to know more about the PCR step, I made a video about that, which I just put in the video description. So you can watch the video where I talk about PCR more in detail later. Finally, I would like to conclude what do we need for the reverse transcription PCR when we go in the lab. So first, of course, we need our RNA sample. Secondly, we need the primers. Further, we need the enzyme called reverse transcriptase, which will generate cDNA out of our RNA fragment. We need dNTPs for the synthesis, of course. And for the polymerase chain reaction, for the PCR, we need the enzyme called polymerase. So in a nutshell, reverse transcription PCR can synthesize DNA fragments out of an RNA template. And with PCR and applying different cycles, we can generate high quantities of the DNA so that it is possible to sequence the genome. That was it for today. I really hope you understood the main points I wanted to show you. And if you have more questions, drop them in the comments. Otherwise, I'm out. Thank you for watching. Bye.
COVID-19 diagnostics. Performing a nasopharyngeal or oropharyngeal swab. It is essential to follow proper infection control measures when samples are collected from patients with a suspected coronavirus infection. Among other things, the examiner should wear an N95 respirator mask, gown, protective goggles, and gloves. To save resources, we will refrain from using each of these items in this video, and instead only show the technical details of sample collection. Let's begin with the nasopharyngeal swab. The patient should be alert and cooperative. Allow them to first blow their nose. The patient should sit in a relaxed and comfortable position during the sample collection process. Tilt the patient's head back slightly and support it with your non-dominant hand. If necessary, lean the patient's head against a wall to minimize jerky movements. Stand slightly offset from the patient to avoid the risk of contamination in case of a sudden cough or sneeze. Be sure to advise the patient of potential discomfort during sample collection and let the patient know that they are welcome to close their eyes. Sample collection should only be completed using sterile swabs made from artificial cotton with flexible plastic shafts. Calcium alginate swabs or swabs with wooden shafts must not be used. These materials can inactivate viral particles or inhibit PCR tests. Furthermore, wooden shafts are more likely to cause patient injury. Hold the swab like a pen between the thumb, index, and middle fingers. Maintain a loose grip that allows the swab to accommodate any resistance encountered during sample collection to reduce the risk of injury. Avoid using the following hand position during sample collection. Start by inserting the swab horizontally into the left or right nostril. Carefully advance the swab while maintaining a course that is close to both the septum and the floor of the nose, parallel to the palate. Do not advance the swab upwards, but instead straight back until resistance is felt as the swab reaches the posterior nasopharynx. When collecting samples from an adult patient, this corresponds to a travel distance of approximately 5 to 6 centimeters or 2 inches. Leave the swab at this depth for a few seconds while gently wiping the wall by twisting the swab shaft. Slowly and gently remove the swab. Immediately insert the swab into the sample collection tube, which may contain 2 to 3 milliliters of viral transport media. If a nasopharyngeal swab cannot be collected for any reason, an oropharyngeal sample can be also collected through the mouth. Again, inform the patient that the smear will feel uncomfortable for a short time and may trigger a gag reflex. Ask the patient to open their mouth wide, protrude their tongue forward, and say "ah." Depress the patient's tongue using a tongue depressor. Gently insert the swab into the oral cavity without touching the lips, teeth, tongue, hard palate, or soft palate. These surfaces should be avoided as they are often colonized by a wide range of microbes. Collect the sample from the back wall of the throat. Twist the swab shaft gently but thoroughly, sampling the oropharyngeal wall. Just as with the nasopharyngeal swab, the oropharyngeal swab should now be placed into the sample collection tube and sent to the laboratory as quickly as possible.
Before starting, make sure your working area is cleaned with RNA Zap. The first step is the homogenization step. Pellet all the bacterial cells growing overnight by centrifuging at 12,000 Gs for 2 minutes. After centrifugation, the Laurier broth supernatant is then decanted. The pellet is resuspended in trizol. Lyse cells by vigorously pipetting a few times. Centrifuge sample at 12,000 G for 10 minutes at 4 degrees Celsius. All nucleic acids and proteins in the supernatant are transferred to a new tube. Take special care because phenol inside trizol can cause the microfuge lid to loosen. Do not leave the microfuge tube containing trizol inverted or laid down sideways, as the solution may leak. Next is the phase separation step. Chloroform is added to the sample. The sample is shaken vigorously and incubated for 3 minutes at room temperature. After that, the sample is centrifuged at 12,000 G for 15 minutes. By the end of the centrifugation, you should be able to see an upper colorless aqueous phase, an interphase, and lower red organic phase. The RNA in the aqueous layer is carefully removed and transferred to a new tube. The next step is the RNA precipitation step. 100% isopropyl alcohol is added and incubated for 10 minutes. Then there is another centrifuge step at 12,000 G for 10 minutes at 4 degrees Celsius. The final step is the RNA wash step. The supernatant is removed and the pellet is washed with 75% ethanol. The microfuge tube is then centrifuged at 7,500 G for 5 minutes at 4 degrees Celsius. The ethanol supernatant is decanted and any residual ethanol may be removed via pipetting. The pellet is air dried for 10 minutes and then resuspended in depth sea water. This concludes our tutorial on RNA isolation.
Thank you for purchase our real-time PCR system. First of all, please turn on the machine. You could find this cable in the carton and connect the instrument and computer by this cable. Now we begin to install the software. First, please copy the software from the USB driver to the PC. Double-click the setup file and next. Install. Finish. Double-click this icon and you can see that's the instrument connected or not. If not, we need to set the IP address. You could click here to find the setting. You should select version 4 to set. And input 192.168.1.5. And the second line will be shown automatically. 192.168, the same as the first line. 1 and 1, and press OK. OK. Now double-click the shortcut icon, and you could find the PC has been connected to the instrument. If you find the PCs is still not connected to the instrument, you should turn off the Wi-Fi. Another important thing is that you need to disable automatic sleep from control panel. By now, the software had been downloaded and you could start your qPCR experiments immediately. Thanks for purchase of launching qPCR system. Today, we will learn how to use optimum design and analysis software. Firstly, let's take a look at how to create a new protocol. Click the New button and enter the Properties page. Here, we can edit the experiment name and select the type of experiment we need to use, and then select the reagent dye to be used, and then we enter the protocol editing page to modify the parameters and the reaction volume of the program. For example, we want to change it to 58 degrees here by input 58 degrees directly. The diamond icon in this step indicates that the current step will collect the forensics collection. After our program is edited, we can click Next to advance the plate editing page. The plate editing page defines the name of the target gene to be tested and the name of the sample placed in each sample well. First, we select the sample well position on the right. For example, if the sample we put in is the first column and the 12th column, then we select the first column and then hold down Ctrl to select the 12th column. After selecting the sample well position, we need to tick the target name and add the target name to the well position. We can modify the target name here. For example, American swine fever virus can be changed to ASFV. Click OK. If we need to detect other fluorescent dyes, we can add another target. Click Add, and for the dye, select Hex, which is a second channel dye, and then tick the check mark in front of the target name. Then, our first row of the wells and the 12th row of the wells will simultaneously detect two target genes corresponding to the two fluorescent dyes, and then we can fill in the sample name of each well below. If we need to add some negative and positive controls, then we can define the type of well. For example, select the first well to change the type. Select from the type drop-down list to represent the negative control, and then select the last well. We expand Type drop-down list, then select plus in the list to represent the positive control. After the target name and sample name of each well had been added, we can directly click Next. Open the Run page to run or save it as a template to facilitate the next run of the same experimental program. We click here to save, and then the template can be saved in a place that is easy to find, such as the desktop. When we open this software next time, we don't need to create a new program from the first step. We could directly click the new drop-down key, select the template file, double-click to open the template, and rename a new experiment. When waiting for the program start to run, the running page will display the estimated remaining time, and the graph of amplification curve will be displayed as below. We can also open another data for analysis when another program is running in real time. In Results Analysis page, we could view the amplification curve and the well-positioned map to zoom in or zoom out the curve. We could also change the curve color for each well to facilitate our observation. We could also modify the position of the threshold line according to our needs. Click on the threshold line and drag up and down to change its position. After we finished the analysis, we can click to save the result and then click Export to export the experimental data in different formats. Or we could choose to analyze negative and positive according to the CT value, and then we can directly export the analysis report in PDF or Word format. Thanks for watching, and if you have any question, please email to us for further help.